turning out, lots of folks. This is the first one of these I've given. It's been 7 months since I showed up back in June. And I thought since we couldn't have a State of the Union address this year, potentially, I'll have a State of the Lab address. So what I'm going to try to do is I'm going to go through a lot of different projects that different folks in the lab are working on, so you can get a sense of what my group's been doing for these last few months, if you've been curious. So first off, I'll introduce the Micronauts, the members of the lab. The first person to join the lab was an undergraduate named Anna Hodge. A lot of you probably remember her over the summer. Really great person, incredibly talented scientist. She's now back in, I think, Middlebury, finishing up her school. She'll be visiting again soon, and she's a great friend of the lab still. She came in and did a lot of cool analyses on this antibiotic data set that I'll present on later that put us really ahead of the game and is allowing us to perhaps push that out of the paper in the next few months. But sadly, she's gone. The next person to join the team was Alex Carr. He rotated in the lab over the summer for a couple months, did a lot of cool work looking at correlation structure in time series and trying to pull out putative interactions that one might be able to test for in the wet lab, developing methods for that. And he went on to work in the Beligo lab, and now he's co-advised between Beligo and myself. Next, Christian Diener showed up, the first postdoc to join the group. I'm very fortunate for Christian to have joined my lab. He's an incredibly experienced computational biologist, had done his PhD at Max Planck and another postdoc in Mexico City, and is now here with us doing really cool work in the microbiome that I'll talk about a little bit later. The second postdoc to join my group, Sushmita Patwardhan. She came here and gave a job talk when I had already shown up. Incredibly talented wet lab scientist, worked in these vents in the ocean, did a lot of cool culturing work, very good at coaxing things to grow that shouldn't grow in the lab. In addition to that, she eats what she kills, in a sense, right? So she's not only generating data, but she's also analyzing that data, and has talent for actually analyzing that data as well, and is building those computational skills in my group as we speak. And we will learn more about her work a little bit later. Connor Hyde is a high school intern who joined the group. Really enthusiastic, really sharp kid. He goes to Gibson Lab, which is this amazing Montessori high school. I wish I had gone to this high school. But they give these kids 2 days a week, full days, to just do whatever they want. And it has to be something formal, but they can do like an internship. So he spends 2 days a week in our group, and he wants to learn how to become a wet lab microbiologist. And we'll have a little bit of what he's doing later on as well. Finally, the last person we have working in the lab right now is Amanda Holmes. Just recently came in, a master's student in the School of Public Health at UW, also has a master's degree in nutrition. She has a lot of experience working in nonprofits and working with sort of clinical studies, writing IRBs, and so on. And she's helping Sushmita and I develop a program to recruit infants and mothers to look at the succession of the microbiome over time. So this is the group, these are the Micronauts. I'm incredibly fortunate to have these folks working with me. Oh, and of course, Jeremy Johnson ties us all together. Keeps us honest, make sure all the T's are dotted in the I, or all the T's are crossed and the I's are dotted. And we have a new coming member soon. Nitin's been very nice to allow us to work in Xanadu River Chamber, but we will be getting our own chamber soon, thanks to the Washington Research Foundation. And so that's very exciting as well. Okay, so I'm going to start off with talking about some of the collaborations that we've developed over the last few months and the fruits of those collaborations that have come out so far. So first off, my training was in the labs of these two guys, and they both were veterans of this sort of building global networks of collaborators type of work. So Jack, during my PhD, I worked on the Earth Microbiome Project, if you're familiar with that. The paper was published last year. Hundreds of people across the planet kind of trying to study everything from the microbes that live at the top of the Himalayas to the bottom of the Marianas Trench, right? Sampling all the Earth's environments and asking what is the steady state normal composition of microbiomes in all these different places all across the world. Kind of getting a baseline, kind of like Darwin on the beetle, right? Just going out, seeing what's there, and getting a baseline. And a similar project in the human gut, Eric Alm's involved with, contributing a lot of computational expertise, but it's the same sort of idea. Hundreds of collaborators getting a baseline for how these systems look, and then building up, to be able to build up of that to do more complicated work. And what I've been interested in for a long time, a lot of my PhD work was looking at perturbations or disturbances. And so I've been interested in building a similar type of global network of collaborators to pull down data sets of applied experimental perturbation on microbial ecosystems, and not just look at the steady state of ecosystems, but what happens when you smack the ecosystem. And so a couple of years ago, at the Ecological Society of America conference, I ran into Jane Rocca and Marie Simonin. Please, Frank, I don't know if I pronounced that correctly. Great postdocs at Duke University, and in collaboration with them and about 6 other scientists around the country, we've begun to pull down hundreds of data sets of these experimental perturbations on microbial ecosystems, calling it the Microbiome Stress Project. We actually had our first paper published this month out in Frontiers in Microbiology, where we do a small meta-analysis of the first few data sets that we pulled down. So we're still cleaning and curating the hundreds of data sets, but for this first little meta-analysis, we had 9 data sets that we chose. These are the distribution across the planet. I'm going to start to read the text. And we had some basic hypotheses or ideas that we wanted to test out of this data set. So, the first objective is you look at one stressor in one environment. Imagine you have, say, antibiotic perturbation, maybe beta-lactam in the human gut across many studies. If you look across many studies, do you see consistent effects of that given perturbation in a given environment? Second objective was to look in one environment across many perturbations. Do we start to see emergent things? Do we see similarities in how given environments microbiota shift in response to just any old perturbation? Then you can do the mirror image of that, which is you pick a perturbation like copper stress and apply it across many different ecosystems and ask, do we see similar phylogenetic structure to the response and so on? And then in the end, right, the overall effect of stress on ecosystems. Are there conserved structural changes in the ecosystem at a kind of high structural level or at a more resolved taxonomic level that you can pull up for when you whack something, what happens? And we had some cartoons of what we thought might happen, but we don't need cartoons because we actually have data. So this is sort of the aggregate of all these different studies. In general, we're looking at alpha diversity now and beta diversity. Alpha diversity is essentially the diversity of the number of species or the evenness of the distribution of abundances of species within a given sample. And beta diversity is showing the differences between samples, the differences between ecosystems. So at the alpha diversity level, generally, if you smack an ecosystem, on average, diversity declines. And this fits well with lots of other stuff in the literature, although we'll add a wrench to that in a bit. Evenness is less clear, so the evenness of distribution of individuals among species is not always changing consistently with disturbance, although in some cases it seems to decline. And here you see some different ways of applying disturbances. It's been a bit of an average here. But if you blow this out into all the different studies and all the different combinations, you actually see a wide diversity of responses. So in some cases, you get a collapse of species diversity. In other cases, you get a huge enrichment in species diversity. And this points to, and agrees with some of my prior work, that a response of a system to disturbance is a non-monotonic function. There's likely a nonlinear function that maps how a system responds to disturbance depending on the frequency of that disturbance, intensity of that disturbance, and maybe even the quality of that disturbance. And that's all stuff that we're moving towards understanding.
So one quick question. So what's the turnover mechanism? So when you stress soil, for example, I'm just assuming how the experiment was done. You incubate soil at higher low temperature and then you do before and after composition analysis? Or
so these are all different studies that were done by different groups, and we're pulling down that open access data. So every study has different methods. In some cases, you go out to a field site and you dump a bunch of diesel fuel onto the soil, right, and you see what happens. In other cases, you take soil from the field into the lab and you do some control perturbation in the lab. But there's a lot of different versions of how these things are done, and I think that'll be important to look at when we're doing our final analysis.
So that's what I'm trying to understand. So what's the turnover mechanism? So because you're just looking at the genomic content, right? Or metagenomic content.
Currently, we have some functional readouts for some of these studies, but mostly that's what we're focusing on.
So unless the cells lyse open or get washed out, or you can't tell between live and dead cells. So how does the composition change is what I'm trying to understand.
I see. So in soils, for example, turnover could be low because you could have things found in clay particles like DNA of dead cells. In the ocean, you'd expect higher turnover, right? Cells are probably turning over much faster. In the gut, very high turnover. So I think your interpretation of the data will change quite a bit depending on that turnover. There's a great researcher named Ken Losey who's done a lot of simulation work looking at residence time in ecosystems and growth rates and turnover, and how that feeds into these diversity responses, structural responses. So I think that work is just beginning to be understood, but it's super cool to think about. One thing I forgot to mention is dispersion increases with disturbance as well. So this is just saying the community becomes more dissimilar from its original state if you smack it, which makes sense. Another couple of patterns that are a little more specific to individual types of communities I'll show now. So for example, looking at the diversity, this is kind of an aggregate version of diversity that includes both richness and evenness. And this is a messy looking scatter plot, but if you fit it on a linear curve, there might be a hump in the middle here. Essentially, a really low diversity community, if you smack it, it is less resistant to disturbance, it seems, right? So it goes further away from the undisturbed community state. And that's perhaps due to functional redundancy is lower in less diverse systems, or who knows what's going on. And even and also on the other end, really, really high diversity communities appear to also have lower resistance to disturbance, potentially. So there might be a sweet spot for resistance to disturbance.
What is this vertical measure?
This is essentially the distance so your resistance, if your resistance is 1.0, if you smack the system, you haven't moved at all in community structure space from the healthy control.
So this is a beta diversity type measure?
Beta diversity, exactly. And if you look in the individual taxon level, you get heterogeneous responses. So for example, in soil ecosystems, if you hit them with copper stress, you see that chloroflexi and acidobacteria increase in abundance, but if you hit sludge, like sewage sludge, with the same copper stress, you see the opposite effect. So we're just beginning to understand these things.
So one more question, sorry. So when you see increase in beta diversity, it's just indicating you're stimulating the low abundance organisms that you couldn't detect earlier, but you detect them because they've expanded in population?
That could be. It could be expansion of low abundance things.
Right?
It could be death. Well, you could have new species coming in, because a lot of these experiments are done in the field.
Systems.
Yeah. So it could be top down, you're killing the most abundant things. It could be bottom up, the low abundance things are blooming, or it could be influx of other species from through dispersal. Again, we don't have absolute control on all these studies as far as the parameters that are going to change. So we're going to have to get more refined in how we stratify the analysis. But ultimately, we want to ask some kind of theoretical ecology questions of, right, for example, there's this hypothesis from Connell in the 1970s that based on life history strategy trade-offs along a diversity gradient, you actually get a unimodal response. That there is a sweet spot of high diversity at intermediate levels of disturbance. So we're trying to explore this process. In addition, the productivity of an ecosystem, or essentially the access to resources, can modulate your response. The sort of growth rate of a urban system can modulate how you respond to disturbance. And that's also something we want to start looking at. But this is just a really quick overview of the beginnings of that collaboration. I'm gonna jump really fast between these here. So sorry if it's information overload. Now we're gonna go into infants. So I think when it comes to the human microbiome and health, there's a window in our lives where it's hugely impactful what happens to our microbiota. There's an ecological succession, and if that goes wrong, there are long-term consequences that last our entire lifespan. And so we're trying to establish different ways of looking at that. So this is work with Sushmita and Amanda and some collaborators in the Swedish system, where we want to recruit infant mother pairs and follow these children over time. Essentially, we really want to understand the mechanisms of ecological colonization and succession, where are the strains coming from? Are they coming from mom, dad, the environment, the dog, so on and so forth? Do you see personalized evolution within these children over time? Are individual diets, maybe breastfed or formula fed, and so on? And these are kind of not well understood processes, and we want to have a baseline study of that. And so how do these transient states impact health? So we know that we have within-person evolution. This is some work I did in my postdoc with Jade Zhao and Tammy Lieberman, where we show that for this one species of Bacteroides fragilis, there's all this evolutionary dynamics occurring in the system. So it seems that they were colonized by a single ancestor about 10 years prior, and you see the rise of these different mutants competing with one another. And here's an example of one sweeping to fixation here, where there's an adaptation in an outer membrane protein that seems to be highly adaptive to this very So we know this is occurring, but we don't really know what the functional impact of this evolution is on the host. In addition, the evolutionary dynamics that we might be able to track, there are ecological dynamics that are incredibly important to health. So for example, these are average time series of children that were vaginally born, and this is an average time series of children that are cesarean born. And you can see in this first year of life, there is a lack of this entire phylum, the Bacteroides phylum. And it's thought that exposure to this lack of exposure to microbial diversity is causal in the slight increase that we see in the prevalence of autoimmune disorders, allergies, asthma, later in life in this cohort of kids. Essentially, if you aren't exposed to this diversity, your immune system starts to overreact to antigens later in life.
What is the light blue also Bacteroides underneath the dark blue? Because that also shows sort of an interesting difference.
It may well be like, this might be Prevotella versus Bacteroides. They're probably different genera within the Bacteroides family. This is from another paper, Yesur et al., that was recently published. But yeah, I believe the phylum is colored by these major colors, and then the subdivision colors are like families within that phylum. So recent paper that came out just showing that your risk factors later in childhood, having been cesarean born, so you seem to be a little more higher risk for obesity, a little more higher risk for things like asthma, and this has been borne out in many different validation studies over the past few years. So one thing we want to do to hit the ground running is it'll take a while to recruit this into another cohort. So just grab a bunch of data in adolescents. So Schmidt is spearheading this effort where we're pulling down all known time series, infant time series, where people have sequenced the microbiota. This is just 23 of them. I think we have 25 to 30 now. And you can see the distribution of time points across these different time series. What we're really looking for is, can we pull up consistencies in the dynamical structure of these time series in people that are cesarean or naturally born, or breastfed, or formula fed, people who live in the developing world versus people who live in the Western world. So we have a pretty good representation as well of different geographic locations to pull these data sets from. And we're collaborating with this group, Commence, this company that has developed probiotics for infants, trying to actually translate some of this insight from the infant microbiome into clinical application. Sadly, they were just defunded this year, so the company is called Ipsing. But Veronica and Mel are still soldiering forth, and we're going to complete this analysis. Okay, and then the last collaboration I'll quickly go through before I get into the meat of like what stories are happening in the lab is with the Price group. So a big question that we have in the microbiome space is, even though we found hundreds and hundreds of associations between health and disease and the microbiome, it's a very chicken or the egg problem. Is the microbiome responding to the host and there's no real feedback there, or do you see changes in the microbiome that are causally driving changes in host health? Big question that we don't really know very well, except for a small number of diseases like Clostridium difficile infection. So you have environmental inputs, you have some filter, and then you have phenotypes, right? We know genotype is a filter, but how much of the microbiome is a filter in this process for these environmental inputs? And there are likely many different types of microbiota, many different community structures that funnel down into a healthy metabolic state for a human. And so maybe it's difficult to characterize a healthy microbiome in the phylogenetic sense, but perhaps in the metabolic sense from the host side, there are ways of characterizing this. So this is work in the Price group that I came in on as I arrived here. So Nathan Price is my faculty mentor and has been very generous with bringing me in on microbiome projects he's working on in his lab, along with Lee Hood here. And this work is really spearheaded by two really excellent postdocs in Nathan's group, Tom and Noah, who you all should know. But the focus of this particular study is to ask this question: how do we define health in the microbiome? So again, we're getting back to alpha diversity, and this is just a basic definition. You can either have richness or evenness. It's generally thought in the field that higher diversity is good. People think that more diversity is better. But I don't think biology usually maximizes things like that. Usually you find optima. And so I would have guessed a priori that there's probably more like an optimal spot for microbial diversity in the gut other than just more is better. So we know, for example, that if you get diarrhea, diversity collapses. Really, really low diversity in the gut. And we also know that the metabolic state of the host is qualitatively worse in people with chronic diarrhea. On the other end of the spectrum, I've seen a few studies showing that constipated individuals either have diversities that are on par with a healthy person or slightly higher. And constipation is also associated with putrefaction of proteins, fermentation of proteins. Usually the gut microbiome likes to break down fiber, but if that fiber gets exhausted and the stool spends too much time in the colon, you get protein putrefaction, and then you get all these metabolic side products that are toxicants to the liver, or to the kidneys that can be produced in carcinogens and so on. So this is sort of the backdrop to Noah and Tom's results. This is the part that I got super excited about because you're looking at individual bacterial genera and metabolites in the blood and the correlations between them. But there's also this overriding correlation with diversity that's very striking. So all of these metabolites are strongly correlated with the Shannon diversity of the microbiome. So for example, there are things that are negatively correlated with diversity up here, positively correlated down here. These are the abundances of these phyla in the gut. So Bacteroides is the most abundant genus, sorry, not phyla, in the gut microbiome. And you see that it is anti-correlated with something like p-creosyl sulfate, which is technically thought to be a bad metabolite, a risk factor for certain diseases. And then you see a group of fermentations that are positively correlated with this. But overall, they were able to get a really great prediction using a small number of metabolites, just I think 11 metabolites. They're able to detect these 11 metabolites in the blood of Arabelle patients and use that to predict the alpha diversity of the gut microbiome. And these metabolites make sense. A lot of them are essentially metabolites you would not find in the bloodstream if not for the activity of the microbiota. So they are derived from the metabolism of microbes. So for example, p-creosyl sulfate, thought to be a bad thing, is positively correlated with alpha diversity, and this is a bile acid that's negatively correlated with alpha diversity. In addition to this training cohort, they also had a validation data set, and they show that you essentially get great prediction of Shannon or of these low diversity quartiles independent of the data set. And this has also been borne out by a few other data sets where several other studies have shown that for a subset of the metabolites that Noah and Tom identified, they're also identified to be correlated with diversity in these other studies. So it seems like a really generalizable phenomenon. And so this is just the prediction in the validation cohort and the correlation in the lasso model between these two different data sets. So this is really cool. And this is kind of one of the first attempts at really bridging the gap between do fluctuations in the microbiome change human health. And I think for our field moving forward, we take it more seriously. This is really the direction we need to go. This is super cool for tying things to health. So for example, here we can really show that Shannon diversity decreases with diarrhea. This is just from questionnaire data, so it's kind of noisy. We still see a nice signal. And even in the predicted metabolite diversity, we see a similar signal. And if you tie this Shannon to different things that we can detect in the questionnaires from the patients, we find that, for example, abdominal pain is negatively associated with diversity, so low diversity, you have higher abdominal pain. Same for diarrhea, these things kind of go hand in hand, but constipation is positively associated with Shannon diversity. So again, we're back to our sweet spot. There may not be a maximization you want, but maybe an optimization. So we're suggesting there's perhaps a Goldilocks zone for alpha diversity in the microbiome, which might be clinically relevant. And if you can detect this through the metabolome, this becomes feasible to be applied in the clinic. Okay. So through the collaborations, now I'm going to talk about the first maturing story out of our group. These are data. Oh, sorry. I changed these slides around, so I'm a little off my flow here. But one question you can ask is, do the good bugs in our microbiota, can they fight back against antibiotics? So antibiotics are great, but they're kind of a scorched earth policy. You're not only killing the pathogen, you're also wiping out all of your indigenous good commensal-like organisms. But is it possible for commensals to fight back against these antibiotic assaults? So Fleming discovered penicillin back in the last century, and it's been a huge boon in medicine, billions of lives saved from this. You used to be able to die from an infected paper cut. That doesn't happen anymore, which is great. But there's been a sort of collateral cost to antibiotics that has gone unappreciated. One thing that became appreciated very quickly was the rise of antibiotic resistance in pathogens, right? So this is a cost of use of antibiotics. Use of antibiotics makes you susceptible to disease as well. A healthy microbiota is actually a barrier against infection. So people who take antibiotics, for example, get infected frequently with Clostridium difficile in the hospital. We see a rise in what are called modern plagues, which usually have to do with the immune system and the overactiveness of the immune system. And this is tied to decreasing diversity in the microbiome and metabolic dysregulation and obesity and things like this. So antibiotic use, diet, and Western lifestyle all sort of come together to deplete microbial diversity, and this is related to these modern plagues. So over the years, over the past few decades, we've seen a rise in these autoimmunity type diseases like diabetes, multiple sclerosis, IBD, asthma, and so on. If you look at a map of antibiotic use and obesity rates in the United States, there is a pretty strong correlation between the two. Who knows if that correlation is not indirect or direct, but these modern plagues are in part seem to be driven by a depletion of microbial diversity. So it's really important to know whether or not the microbiota can find mechanisms to resist antibiotics. So how do they mitigate this damage? So when I was in my last year in the Gibbons Lab, I was collecting some mouse data and was looking at other studies that had thought about these types of processes. And one was this study from Pat Schloss's group, where they're looking at colonization resistance and antibiotic treatment. So if you crash the ecology of a mouse gut and you apply Clostridium difficile spores, you get colonization of this pathogen. So it opens this mouse up to colonization and they die from this pathogen. But if you have a nice healthy mouse with a nice healthy microbiota, you give them Clostridium difficile spores, nothing happens. They're resistant to the invasion and they survive. And the antibiotic that was the most effective at pushing mice towards being susceptible to colonization was the cefepime antibiotic, which is a beta-lactam, very broad spectrum antibiotic that kind of knocks out most things in the system. So this seemed like an interesting antibiotic to zoom in on, but if you look closely at that paper, they didn't actually show the microbial community structure for these antibiotic-perturbed mice on a mouse-by-mouse basis. They just showed averaged error bars. But if you looked at, they had some machine learning, a random forest model that they ran to predict colonization of C. diff from the microbiota, and what you find, so for example, no antibiotics, you essentially get no colonization of C. diff, but you also predict no colonization. But for cefepime, you see that there's a subset of the population that is neither colonized or predicted to be colonized by Clostridium difficile, which to me means that there's a subset of the population that does not respond to antibiotics. There's a subset of the population that maintains some structure to their community that's very similar to the control community that protected it from this perturbation. And this is an uncharacterized phenomenon. No one really has discussed this in the literature. So this was a hypothesis we wanted to test in a mouse study. So what is the difference between these communities that allow for resistance to this perturbation? So first off, we had this duration of exposure experiment. So in this study, they actually tried different concentrations of antibiotic, and you see this sort of heterogeneous effect across different concentrations. We wanted to ask whether duration was a factor in manipulating the ability of the community to resist antibiotics or not. So we had 4 different durations of exposure to the highest concentration of cefepime that Pat Schloss used. We really wanted to hit the system really hard, but for different periods of time, 2, 4, 8, 16 days. And this work is spearheaded by Christian. Anna did some work on this over the summer, and then a student in Gibbons Lab, Sean Carney, helped out a bit with the mouse work in the lab. So if you look at, these are the results in a principal coordinate analysis, essentially. These, the green and the blue mice were both hit with antibiotics. They were hit hard with really high concentration of antibiotics. The red are the controls that received no treatment. And there's an obvious heterogeneity here. Some of the mice responded to antibiotics, and some of the mice didn't respond so much to antibiotics. So there might be some intermediate phenotype to some extent. These symbols are just the number of days after antibiotic treatment for that mouse, for that sample. We actually zoom into the community structure. You can look at this is sort of the phyla that were most abundant in the control control that received no antibiotics, you see what you expect. Bacteroides and Firmicutes are the most abundant phyla in a nice healthy mouse gut. But we see the segregation of these two populations in non-responders and responders. The non-responders, they look indistinguishable from the controls. There's not really much of a significant difference between the structure of these communities and this community here. The responders, total collapse of the community, completely upturning everything. So now we're dominated totally by proteobacteria and cyanobacteria, which has been seen before.
Sean, just a quick question. Did you give antibiotics again to the non-responders? I'm just curious, are they persistent non-responders or were they just?
That's a good question.
One time?
We did not do that particular experiment with the same mice, but we did do a second follow-up experiment to try to recapitulate our observation here. But that would be an interesting one to do in the future. So if you look at the different durations, there was no significant difference in the frequency of the non-responder phenotype depending on duration of exposure. So duration had no effect on whether or not you were a responder or a non-responder, which is sort of surprising. You might expect that non-responders would be depleted with longer and longer durations of exposure. So at first I thought this was a mistake. I tried to write it off, but we ran another experiment and we saw something similar. So 6 out of 16 mice here did not respond to antibiotics. If you think about, the clinical implications of this, right, in the human population, we see a loss of diversity from antibiotic exposure is potentially implicated in these autoimmune disorders. So if you look at the sort of the extinction rates of these different groups, in the controls and non-responders, there's not that many species lost from the beginning to the end of the experiment, but there's a definite significant uptick in the loss or extinction of species within a given mouse when you are treated to respond to antibiotics. So these non-responders are protected somehow from this loss of diversity, both from a collapse of the structure of the community and a loss of alpha diversity of the system. So this is now a time series of days post-treatment, and you can see very clearly that the mice that respond to antibiotics have a sharp decrease in their alpha diversity and a slow recovery over time, and the non-responders have some sort of intermediate phenotype here in between. And if you look at long-term resilience,
How do these individual mice vary? Are they genetically identical, or are they? Do they initially have an identical?
Yes, they were initially identical mice. They're all black 6 female mice from the same birth cohort, housed together before the study, and then before we started the study, singly housing them. So one thing about mice is they're coprophagic, they eat their own poop. So if you house them together in the same cage, they're all going to eat each other's turds, and their microbiome is going to average out. So we really wanted to get individual statistics on that, what each individual mouse does. So at the beginning, we homogenized them by co-housing, and then we separated them prior to the study. One important observation as well, in addition to this alpha diversity loss, is for some of these mice that respond to antibiotics, you see a flip in the normal structure after recovery, where Firmicutes become the dominant phyla and Bacteroides are less dominant. And in many of the mice, you never get a recovery of this crucial phylum, this entire phylum of organisms in this mouse gut. So there seems to be an extinction event that happens in some of these mice. Normally when they're co-housed, this wouldn't happen because these mice would actually recolonize these with Bacteroides. So a loss of resilience in the responders, loss of diversity, and a collapse of the ecosystem. So we redid this because I thought, something might have gone wrong, who knows what happened. So we redid a whole other experiment with another 16 mice. And we actually had a dietary treatment, but I don't have time to talk about today. But essentially, 4 out of the 16 mice were non-responders. And we get a similar sort of pattern where the non-responders are pretty close to the controls, a little bit of an intermediate phenotype, and a collapse of the community for the responders. You could ask, this is all relative abundances, what's happening to the total biomass in the community, right? So if you measure by qPCR the total copy numbers of 16S genes in these different samples, in the responders, biomass pretty much collapses to 0. The biomass of the system, all the microbial biomass goes away. But in the non-responders and the controls, it remains quite high, although there's a little bit of an intermediate phenotype here with the non-responders. And if you look at the actual, what are we sequencing when we look at these? Yes?
Oh, just a quick question. Has this been looked at in humans? Do you have a similar responder, non-responder effect in humans that's been observed?
Usually non-responder phenotypes are downplayed. In most of the literature, if you search, you can kind of find it. So in humans, you do see heterogeneous responses. So you see milder responses versus really strong responses to a given antibiotic. There's only been a few studies where they've given antibiotics to healthy people because I guess that takes, it's hard to get the IRB for that. But there was a study in the UK and Sweden where they do that, and they do see some heterogeneity there. So what ends up happening when you sequence a community that has lost all of its bacterial biomass is you essentially are sequencing the mitochondria of the host organism and the chloroplasts of the diet. So you're just sequencing the chloroplasts in the food and the mitochondria in the host gut. So this entire community that we see in relative abundance form is sort of an artifact of the fact that all the biomass has gone away, which is kind of, you can almost use the frequency of mitochondria and chloroplasts, at least in mice data, as a proxy for the biomass in the gut. So what could be going on? The community structure is not that different, so we thought, okay, gene expression might be driving this. There might be some sort of global community gene expression response that might be driving this effect. So this is where Christian, in the wet lab, which is not his usual place, but he did a great job extracting RNA from some of these responder, non-responder mice and controls. And essentially, this is just a volcano plot of the data. I don't want you to see too much in it because it's kind of a mess. But essentially, day 20 and 25 is all pre-antibiotics. So we had the non-responders and controls on these days prior to antibiotic exposure as sort of negative controls. We don't expect that many significant hits between those groups. And indeed, we only get about 5%, after FDR correction, about 5% of things are significant in these 2 days. But on day 29, which is after antibiotic exposure, in these mice that are non-responders that still have high biomass and the community structure looks identical to the controls, 25% of all the genes in the system are differentially expressed. There is quite a strong response, and on a PCA, that's kind of how it looks. So a very significant difference between the controls and the non-responders in gene expression, at least, following antibiotic application. So you actually zoom in onto which genes these are. One of the biggest signals we find are in these virulence, disease, and defense genes, which you might expect. Generally, we see an upregulation of ABC transporters, efflux transporters. So there's a global upregulation of these efflux transporters, which are very general mechanisms for resisting antibiotics across all species in the gut system. So this was a very strong signal. Again, these are kind of antibiotic resistance genes that are upregulated in the non-responders versus the controls. But in addition to this antimicrobial resistance response, we also see kind of what seems to be a global response in growth rate downturn. So acetyl-CoA synthesis is sort of a central point in growth, and this seems to be turned way down in the non-responders compared to the controls. We see potentially a reduced growth rate. We also see motility is much reduced, so the flagellar apparatus is downregulated in these non-responders. And we see other patterns that make sense, like this TON-B system is actually highly coupled to efflux transporters. If you knock this gene out, efflux doesn't work anymore. And this particular gene is upregulated in these non-responders. So this is sort of the story what we want to tell, where you have an initial community with some sort of transcriptional program. You smack it with antibiotics, and some subset of that community is able to rescue itself through its downregulation of the growth and upregulation of antimicrobial resistance. And this is through growth and efflux. And it protects the community from both diversity loss and from biomass collapse.
So two questions. First, have you looked at the baseline diversity and the ratios among the communities? Because even though the diversity could be the same, but the ratios matter. Which population over-dominated at baseline and in the responders versus non-responders? And the second question, what tissue this gene expression came from?
What tissue? Okay, so for the first question, I'm not sure I understand your question.
The first question is like, how did the ratios of communities look like at baseline in non-responders versus responders? Because even though the diversity index was the same for all groups, but maybe the ratios of communities matter at baseline. Maybe
At the gene structure level or the gene expression level?
At the diversity level.
At the diversity level, there's really no significant difference between the two before antibiotics. At the community structure level, there are probably some subtle differences between the groups, but I think that's hard to know if that's a small number of statistics. Each mice has a slightly different microbiome thing, or if that's truly an effect. But it's if it's there, it's very, very subtle.
My concern is like even though the diversity is still the same, but they might be different in ratios. Like we have 70% of the ratio of the population belonging to this specific phylum versus the other phylum in the other group. So
rather than looking at relative abundance, just looking at ratios between two organisms as
it's
a
just like
proxy.
ratios or relative abundance.
You
Because
showed data.
diversity talks about types of populations. It doesn't talk about the ratio of populations, or relative ratio of populations.
Well, we are working in relative abundance currently. I'm still struggling to see exactly what you're saying. Like you can do something like isometric log ratio transforms to relative abundance data to turn it into something that can be used in normal Euclidean space or statistics, which is actually log ratios of relative abundances. I don't know if that's what you're talking about. But maybe in the interest of time, I'll skip ahead, we can talk after. Your second question.
Where does the gene expression came from?
This is whole stool pellets, so little mouse poops, whole thing ground up, all the RNA. Okay.
Is there time for one more question? Or?
Yeah. I mean, yeah, let's do it.
The mechanism of resistance for beta-lactam is very well established. And I can see that being something that sweeps through the population, the gene sweeps through the population through plasmid vectors and so on. But here you're claiming efflux pump expression as means for resistance, which I'm not too familiar with in context of beta-lactams.
It is involved beta-lactam.
Efflux.
Yes,
And is that plasmid encoded or is it?
both chromosomally and
So you
plasmid.
should
But
be
most,
able to tell that, right, from your data, if it's swept through the population.
we might be able to.
Same-
We might be able to. We should look at that. We could look at like the heterozygosity of a given gene at the sequence level and see like if the diversity of that gene drops after antibiotics. That might suggest a sweep. So we could look at that.
For efflux pump, you would need a sweep. If it is a enzyme that is secreted out to integrate the antibiotic, then you don't need a sweep.
Right.
You could have complicated.
Beta-lactamases, for example. But beta-lactamases we didn't see a strong signal for. But most organisms have these efflux pumps, right? Most organisms have some subset of these ABC transporters and efflux pumps, but they're just general mechanisms for resistance, not only antibiotics, but heavy metals and what have you. So we don't quite know the exact story yet, but so this is a little preliminary, but I think it's a cool story.
So can you assign the expressed resistance genes to species based on sequence?
I believe, yes.
Or?
I believe we can. Christian can answer that better than I could probably.
Yeah, I mean, genes is also pretty complicated if you talk in the sense of metatranscriptome, right? Because actually here what we did is not really genes, it's like functional groups, because like if you look at genes, you usually see that they're almost specific to particular samples, they also depend on the and stuff. It's like it's a bit like on a coarse level, like if you have the same like the same function, you basically group like the groups. Yeah. Because in that way you can say that the majority of functions depend on all the samples, you can actually do statistics of them if they're all specific to samples. So those are like functional groups, not like genes and like classes.
All right, I'm going to keep running ahead. Sure. What response genes? Sorry. I see. From my understanding, you can correct me if I'm wrong, but I think the vast majority of the reads are microbial origin, so we can't really find that many host RNAs, I would guess, in these data.
Yeah, it's I mean, we do ribosomal deletion as well. Anyways, so, and then we actually filter out all the reads that come from mice or anything that's actually on the archaea.
That could be interesting.
That is about like 20% max.
Cool. Maybe we should look at that. All right, I'm gonna keep running. So that was like the first kind of almost finished story that hopefully we'll push out in the next couple of months from the group. Now I'll give you a little preview of the first 6 to 12 months, or for the next 6 to 12 months, and hopefully have 5 minutes left over for questions if we're lucky. Okay, so one cool, I wanted to include everybody in this presentation. So Connor has been recently working on computation experiments between microbes in the lab. So one really big challenge in understanding ecological systems and engineering them is to actually understand what are the pairwise interactions and how do we sort of pull apart the system. There's a cool pattern we see in the data where Prevotella and Bacteroides are two genera that seem to inhabit the same niche in different people. This is a Disney of the microbiomes of all the people in Aryvale. And this is the Prevotella group and the Bacteroides group. One excludes the other. It seems to be very difficult for one to invade the other, but we don't understand the mechanism underlying that interaction. So we're starting to work with groups at UW, both Paula's lab, where we have strains isolated. And Shushmita is also kind of mentoring Connor in this. And Alex is developing computational tools, or has developed some computational tools to pull hypotheses out of omics data sets that can then be used to inform the lab work. So we're trying to do this type of stuff. Annie's going to be involved to be able to grow these guys under like gut-like conditions.
Would it make sense to do the Disney excluding Prevotella and Bacteroides?
Oh yeah, we've sliced and diced in all the different ways.
But otherwise, it's not really clear what point you're making with this, other than to say that Disney is able to separate on data that it was given. But if you exclude it, and you saw clusters in the Disney data that then correspond with the colors of Bacteroides and Prevotella, I'm not sure. Disney's so hard to interpret.
Oh, well, like, even if you just look at the abundances. When Prevotella is abundant, there's like 0 Bacteroides. When Bacteroides is abundant, there's like 0 Prevotella. Not 0, but it's like, they seem to be mutually exclusive to some extent. And it's maybe I used the wrong plot to show it, but essentially it's very, very clear to see. If I showed you the stack bar plots, for example. This is the bimodal abundance distribution for Prevotella, essentially. You're either almost near 0 or you're quite abundant in the population.
What are the lines?
These are connecting time points. These are the same person over time. So you can see some transitions between different places in this space. Okay, I'm going to keep running. But another thing we want to do are these ex vivo experiments. So we're going to have Annie soon. Tom, a good friend of mine from the Alma Lab, developed this type of experiment where cerebrate organisms A and B may or may not have been me, and we take poop from these individuals and we can culture them in the anaerobic chamber. We can add very specific compounds and ask what metabolites are produced from those compounds. So in this example, you add inulin to person A's microbiome here, you can see it very quickly is consumed within 10 hours, it's depleted within the incubation. And after 24 hours, if you measure some really important metabolite like short-chain fatty acids, which are involved in anti-inflammatory responses in humans and are generally thought to be good for you, we find that for the very exact same inulin input, this fiber, you get very different short-chain fatty acid profiles. So person A produces a lot more acetate and butyrate, and person B produces a lot more propionate. And the blue is just showing if you add PBS without anything in it. The stool has a bunch of nutrients in it already, so it's going to be producing stuff anyway. Alice is doing some cool stuff. I'm going to kind of run through it. We're writing a perspective paper. I've been sort of mad about how correlation analyses are used in microbial ecology. So we're doing sort of a pedantic, perhaps, perspective piece where we're showing how compositionality, for example, like absolute abundances to relative abundances, can induce phantom correlations. Things like periodic signals are often ignored. So often in ocean time series, we find that there's a periodic signal on about a year time scale, where organisms that live together in the winter versus the summer kind of rise and fall together. But within, like, for example, Vibrio that are more abundant in the summer, within that group, there are all these Vibrio that compete. So you see anti-correlations within the group. But the overriding seasonal signal kind of abolishes the negative interaction when you do the correlation analysis. So for example, you could take these time series, you could decompose them into frequency space and see that, okay, you have 2 different frequencies, low and high, and you can see that in one they're positively correlated, in the other they're negatively correlated. So using more information than just pure correlation to sort of dig a little deeper and get a little better information on what's going on in your systems. And the final thing I want to talk about, which I'm super excited about, is some work that Christian's been working on, and OHAD is also involved from Maryville, where we're trying to, so back, Tom and Noah have some really cool data linking the microbiome to health, but we still don't have arrows. We still don't know what causes what's the directionality. If this thing goes up, what happens to the other thing next? In the Aryville data set, if you look at when the microbiome is measured versus when the metabolome is measured, you end up getting a distribution that looks something like this, where here the microbiome is measured first, and here the metabolome is measured first. And we can use these data to our benefit. We can use this asymmetry to our benefit when we segregate our analysis to try to look for associations. So for example, in this particular pair, Ruminococcus and this metabolite, we see that there is a significant correlation when microbiome may be first, metabolite second, but that correlation is weaker or absent in the other direction. And we want to do here's an example in the other direction. We want to really do a global analysis to try to build these directed networks, which are sort of putative interaction networks or putative causal networks that can pay perhaps better direct experimental work or follow-up studies, and actually help you to engineer the structure of the microbiome. So if you knew that adding in this metabolite can increase the abundance of, what's a good example? Adding in this metabolite increases the abundance of this organism, and maybe this is butyrate, which is a good metabolite that you want. So if you feed somebody fiber, you get butyrate out. Knowing those chains would be super useful for engineering the metabolism and the structure of the microbiome. And with that, I believe the state of the lab is fairly strong, and my New Year's resolution is to quickly establish ISB as a center for microbiome excellence. With that, thanks to you all for listening. Thanks to the funders and all the people